big questions. So this is the way I try to tell a lay audience why active matter is fun. And we're going to look at lots of movies, and that will give you some idea of what the active matter systems are, the main experimental systems, and the sort of questions that we want to ask about them. And then I think most of the rest of today will be spent doing some sort of background, which we're going to need to understand the rest of this. And first of all, I'm going to talk about hydrodynamics at low Reynolds number, overdamped systems. We've heard about those quite a lot in the last couple of days. And this is going to be relevant for the active matter, but it's also useful all the way across biology. So hopefully it'll be useful for people who are not directly working on active matter. Hello? What's wrong?
You have to press the
button. Oh, I haven't pressed the button. I thought I pressed
it.
Yes. Oh, okay. Yeah. And then we're going to talk about the idea of nematic liquid crystals, and in particular, topological defects. Because I think one of the really big things about active matter is that you find topological defects, and in particular, self-propelled topological defects. And people are now finding these things in biological systems, although the jury is still out, perhaps, just how important they are, but it's quite fun trying to find out. So then I'm going to move on to active matters properly and talk about active turbulence and these defects. And then from 6 to 8 is sort of what I feel like doing at the time, actually using this stuff and using applications. Okay, so let's get started. What is active matter? Well, we've really heard from Ralph what active matter is. And to me, the way I like to describe it is it's systems which are continually taking energy from their surroundings and using it to do something like move, using it to do work. And they do that as individual particles. So it's not like taking a fluid and putting in energy by shearing the fluid. It's that each particle in that fluid takes energy from its surroundings and is able, therefore, to do work, often to move. And so these systems are out of equilibrium, okay? We're out of equilibrium. Human beings are out of equilibrium. All living systems are out of equilibrium because we're alive. We can actually, most of the time, I know late at night it gets a bit more tricky, but most of the time we can use energy to do things either on a microscopic level in individual cells or just on a macroscopic level of moving around and listening to talks. Now, these active systems, you find them across all sorts of length scales. Inside a cell, well, you've got to have a way of moving things around inside cells, of doing work inside cells. And the things that do that are molecular motors, and we'll think a bit about those. And then on a cellular level, a lot of experiments on active matter deal with these tiny swimming things, with microswimmers, or with eukaryotic cells, squidgy cells. And then we have fabricated active matter, which is really very simple compared to biological active matter. And mostly there, people worry about active colloids. And then all the way up to macro scales. We'll see some movies in a minute of fish and birds. And on the whole, we concentrate on the tiny things, and that's because they're easiest. You don't have all sorts of Garth-like things worrying to think about things to decide what they want to do. So, why can you move? Why can you do anything? How does your cells work? We need some sort of engines to move things around in the cell. For example, to move proteins around in the cell. Okay? And certainly when I was at school, it was, well, how do you move muscles, right? And I never asked the right question, which was, okay, so muscles move, but what makes them move? But now, I'm sure you are much more used to thinking of things on a much smaller length scale. You can worry about cells. What is it in our cells that make us able to live? What are the engines of the cell? Well, here's one of them. This is kinesin, and this is my absolutely favorite movie of all time. Okay, this is what happens inside a cell. Inside a cell, you lay down tracks, the cell lays down tracks, and then this little pair of feet here is a kinesin motor which walks along the tracks and carries behind it some sort of cargo in a sort of vacuole, which it essentially is a bit like a backpack. Let's just play that again. So for a start, why do we worry about having tracks? Okay, why does the cell bother to lay down tracks? And the answer to that is that we're very small here. We're at nanometers, and so thermal fluctuations, the Brownian motion we've been hearing about, is very, very important. And so if you just had a molecular motor sort of just trying to get anywhere without something to hang on to, it would just get wafted away by all the molecules, all the other proteins, all the junk in the cell hitting it. So you have to have these tracks. So that's a wonderful question to start with, right? How on earth does the cell know how to lay down the tracks and how to put the motors on the right tracks and how to make them move in the right direction and how to get the right cargo in the cell? That's a control theory question, and I believe that very, very little is known about that. Even then, how do these things move? This is a protein. This molecular motor is a protein. It really does look, I mean, this is a computer animation, but it really does look like a pair of feet moving forwards like this. It's not quite so regular. It's actually much more stochastic. It doesn't quite, do it quite so efficiently, but it does manage to move along these tracks. And basically, it's because it's a protein with a very clever structure. What happens is that the thing gets a molecule of ATP from the surroundings in the back foot. That means that it can detach from the track. So you've got this head, which is sort of waving around with Brownian motion. But the tethering the two feet together, acts pretty much like a spring. And so the back foot tends to be pulled forwards because of the structure of that protein to lie in front of the other foot. And so it's more likely to grab onto the track in front. And then it releases a molecule of the other one, TP, I think. And then that allows the back foot to be ready to get a fuel molecule and start again. So this is an amazing design. Nature has somehow managed to design this thing that can walk along tracks. Just so I'm not totally kidding, because I mean, you look at this and you think, really? But it's right, it's true, you get used to it. Okay, here are the two heads which are stuck on the track. These are light microscopy pictures, and as a function of time, you can see these things moving forwards. Okay, and they do have movies, but it's really quite hard to sort of catch them on the screen. Pretty amazing, huh? Here's another one, another sort of molecular motor. This is a bacteria flagella motor, and this is how bacteria swim. It sits here between the body of the bacterium and the tail. So it's here and across the cell membrane. And it sort of looks like this. The sort of is that, to some extent, this is an artist's impression, but people have some idea of how it fits together. All these different bits are different sorts of proteins, and it's driven by an ion current across the membrane. Here's the flagellum sticking out here. Okay, so this thing here is across this little membrane there. So look at the size, right? 27 nanometers. And that's made up of lots of different proteins. How on earth does nature manage to put them together? How do you build something like that? We can't do it, okay? Nature obviously can do it because it does. And then it has to mend it because this thing's going around pretty fast. And so friction is going to knock the proteins out. Things are going to go wrong. And so it has to be repaired. And so you have an absolutely amazing system here where the active self-assembly is giving you something like that. Oh, this is again an artist's impression about how it might form. Okay, but some really nice problems here for you, trying to work out actually how this does work, actually how active self-assembly can give you this sort of picture. And of course, the reason these questions are starting to be asked is because of the improved microscopy and the improved nanoscale technology, which means we can start to look at motors like this and start to try and understand how they work. So this is very much a best guess. So what can we do? Can we fabricate anything like this? So this is maybe the best, certainly a couple of years ago, is the best sort of active motor. These are little discs with these gears, essentially, okay? Length scales about 60 microns. And these dots in the background are bacteria. Yep. The bacteria are pushing the rotor around, really because of the interactions when they hit the thing. It's okay, it's not perpetual motion. It's not perpetual motion because the bacteria are eating stuff, so what they're really doing is changing chemical energy into mechanical energy. Okay, and you can create beautiful things like this. And these are nanoscopic machines. These are clever experiments, but compare them to those molecular motors in the cell. Okay, we've got a long way to go to try and do anything as well as nature can do it. This is, human version of active self-assembly. If you put creatures which are self-propelled, so these are these little kids' toys, okay, and you put them on a surface, and you put these V shapes on the surface. Whoops. Thought this was a movie, but maybe it isn't a movie. No, I think it's just what happens at the end. And what you can see is at the end, what you end up with is everything organizes itself onto this side of the barrier. And the reason it does that is because of the shape of the barrier. The shape of the barrier breaks the symmetry. If one of these creatures hits there, it can slide easily to this side. If it comes in this direction, it has trouble getting around this corner, and therefore it tends to bounce back. This is exactly the same thing with bacteria. Here's the line of V-shaped obstacles, much tinier ones now. And this is a thing which contains bacteria. And as a function of time, the bacteria, which show up as the fluorescent white bit, self-organize themselves to be on this side of the barrier. So active self-assembly. But again, we're a long way off doing anything that nature can do. These are active colloids. People are trying to make active systems. The way they make these is that they're colloids, and there's a little bit of hematite stuck on the end of a colloid, and then they are put in a suspension with hydrogen peroxide in it. If you put on light, this hematite reacts with the hydrogen peroxide, and essentially they behave like little rockets. I think it creates bubbles, and therefore they behave like little rockets, and they become self-propelled when you have lights on. And when they are self-propelled, when they're acting as self-propelled colloids, what happens is that they sort of move towards each other and they get stuck in these rafts. Again, active self-assembly, but a rather simple version. Just to check it works, in a minute they're going to turn the light off, and then the Brownian motion is going to break up the rafts. There we go. And this counts as an active system because the light is addressing each of these particles individually. Okay, so what we are going to concentrate most in this talk is now, let's take these active systems and let's put them together. So we've got a dense collection of active particles. What I've got here is a dense two-dimensional suspension of these microswimmers. These guys are E. coli, I think, although I must say I probably just got that from somewhere. But this is real experiments, and this is what you see. Looks turbulent, yeah? Looks chaotic. The flow fields look chaotic. So what we're doing here is now statistical physics, right? Because statistical physics is what happens when you put loads of particles together. But these are different from normal Boltzmann-type statistical physics because there we're in equilibrium, we understand how to do it, we know about Boltzmann distributions. Here, each of those particles is out of equilibrium. When we put them all together, what do? Why do we get this sort of turbulent-like state? I mean, it's weird because they're tiny, right? Tiny means low Reynolds number. And at low Reynolds number, I'll come back to this in a minute, things normally flow very smoothly and calmly. Turbulence is high Reynolds number, high velocities. Okay, so that's something to explain. Let's see what other of these active systems do when you put them together. These are cells. This is a two-dimensional layer of eukaryotic cells. Eukaryotic cells are squishy cells. This is the sort of thing you have on the lining of your stomach, okay? And these are an active system. They're alive. So they're always taking energy from their surroundings, and then somehow that energy is being transduced into motion. And again, it's pretty chaotic motion. Is it the same as the microswimmers? You know, is there any underlying physical principle we can sensibly talk about, or is it just something completely different? Let's look at the macroscopic ones. I mean, here I understand nothing, and it's just such a beautiful problem. Okay, that's a shoal of fish. That is apparently a seal, right? I showed the people in the Netherlands who know about seals, and they said it was a seal. I'm sure it's not a shark because of what these guys are doing, okay? I thought it was probably a porpoise or a dolphin, but it's a seal apparently, and okay, and it wants its dinner. But there's some sort of collective behavior going on here. Collective behavior of active systems. And I'd love to be able to understand why this happens, but I have no idea how you'd model something like that. There are people who've tried to do collective animal models, and very interesting they are too, but I don't think they can explain what's going on here, or indeed, oh dear, that's all right. This is the other one. These beautiful flocks of starlings. Are you going to move? No, sorry, I've got a movie there. Meant to be a movie? No, it's not a movie. All right, but if you look at the flocks of starlings, again, you get these swirly patterns, sort of two-dimensional sheets which intertwine with each other. These are active systems, but they're much more complicated active systems, because as well as just you're putting in energy and these things are moving, they can think on the way. They can probably do things like look at their neighbors and see if there's a difference. And there's been a lot of work trying to understand this, and I believe that nobody yet really understands what's going on in these systems. Okay, so that's why we study active matter. So do people have questions? I'm really pleased about questions, if anyone's got them. Any questions? No? Right. Please interrupt me, all right, because it's much more important that you understand what I'm on about. So when I don't explain things properly, just tell me. Okay? So this is why we study active matter, at least why I study active matter. Biological systems do physics. And therefore, maybe physical principles are going to tell us useful things about biology. There's, of course, a long history of that, from the invention of DNA, ideas about Turing patterns, things like that. And biologists like detail and physicists like generic things, and putting them together, I think, could be very powerful. From a technological point of view, the cell is an amazing engine. Can we exploit the ideas that you have in biology to create and design better engines? And then very much following on from Ralph's talk, this is non-equilibrium statistical physics. These are experimental systems which allow us to test the ideas of non-equilibrium statistical physics. Okay, so that was sort of the party political broadcast on behalf of active matter. Let's actually do some proper science now. I'm going to do it this way around, I think. I'm going to talk about low Reynolds number hydrodynamics. And where I want to get to is to have you understand what sort of flow fields you get around swimmers. As swimmers move, these little micro swimmers, they set up a surrounding flow field. Okay, what does it look like? That's where we're going to get to, but hopefully we'll do some other things on the way. So that's the Navier-Stokes equation. Okay, have we got anyone here who hasn't seen Navier-Stokes before? It's right, you're allowed to volunteer. Yeah, just one or two of you, right? This is the equation which tells me how fluids move, how things like water or gases move. And it's a continuum equation, so I don't care about molecules. All right, I just think of it as a continuum with a certain viscosity and a certain density. So this is density. V is the velocity, time derivative, v dot grad v minus the gradient of the pressure. Pressure gradients are going to give me a force which will make this fluid move. This is viscosity. Okay, viscosity is essentially if you try to push a fluid, eventually it will stop moving because of gradients will damp out and then any forces you put on. That's a pretty amazing equation because it can describe this, what's happening in clouds. It can describe waterfalls. It can describe tea, very important in Britain. It can describe that, wine, glasses, which is even more important in Britain. Okay, and all the way down to bacteria. That's pretty amazing, isn't it? Right, you know, this equation, this is why I'm a theoretical physicist, because you can write down that and you can understand things about so many different things. Okay, so the important thing about Navier-Stokes, one important thing, is that these guys on this side are called the inertial terms, and those there are the viscous terms. If you're swimming in a swimming pool and you stop moving, because you've got momentum, you will keep going. And that's what is described by these inertial terms. If you just had conservation of momentum, the inertial terms would be the important ones. But you know you don't keep going forever, and that's because of these terms here, which are the viscous terms. The viscosity will eventually slow you down and bring you to a halt. So what we'd like to do is look at the relative magnitude of the inertial terms and the viscous terms. And the way to do that is to non-dimensionalize the equations, to rewrite them in terms of a velocity, which is our velocity scaled by some sort of typical velocity in the system, and our positions scaled by some sort of length, L0. All right, so I've done that for you. I've written everything in terms of dimensionless variables, and what comes out is that I end up with an inertial term with nothing out the front. And here's my viscous term, del squared V. And in front of it is this parameter here, well, a set of parameters, okay? Viscosity, length scale, velocity scale, density. And this thing here is 1 over the Reynolds number. Because if I look at the ratio of the inertial response to the viscous response, if I look at the ratio of this bracket to del squared V, this is what comes out. Sorry, 1 over that is what comes out, okay? And this is the Reynolds number. If the Reynolds number is big, it means the inertial term is important. It means there's a lot of momentum around, and it tends to be true at high velocities and large length scales. So these guys down here are high Reynolds number, lots of vorticity and turbulence. If the Reynolds number is small, that's small velocities and small length scales, microscopic things, then everything's very calm, and things flow through pipes in nice straight lines, and you have real trouble trying to mix things up. So what is it for our swimmers? I'm thinking now of bacteria. What sort of numbers do we have for bacteria? Okay. Typical length scale for a bacteria, maybe 10 to the minus 6, a micron. Actually, that's a bit small. It's probably nearer 10 to the minus 4, but it's something pretty tiny. These things tend to go at 1 micron per second, 10 to the minus 6 in sensible units. And then you just have to look up the ratio of density and viscosity, and it turns out that this thing is a 10 to the 6 on the bottom. So the Reynolds number is about 10 to the minus 6. I've probably overestimated its tininess. It's probably 10 to the minus 4 is probably a better estimate, okay? But it's tiny. It's a lot less than 1. We're absolutely in the low Reynolds number limit. And what you can do in the low Reynolds number limit is forget completely about these inertial terms, get rid of them, and you end up with this, which is called the Stokes equation. All right? So okay, so Stokes equation, so viscosity wins. So this is my favorite movie. This is a very, very, I hope this one is going to work, actually. I should be pretty fed up if it doesn't. Right. Okay, so this shows you what fluid mechanics is like at 0 Reynolds number, at tiny length scales and high viscosities. Now, quite reasonably, you might be saying, this is not tiny length scales, that's someone's hand. This is not microns, this is centimeters. So why am I okay? Why is this still low Reynolds number? Anybody like to hazard a guess? Yeah, exactly. This is not water, this is glycerol. So it's got a very high viscosity. So high viscosity at normal length scales and water at tiny length scales gives me the same very small Reynolds number. Now you have to count. All right, this is like a nursery class, really. You have to count. I think it's how many times they're going round, very slowly. All right, that's just colored dye. 1, 2. It goes on a bit, this movie. Normally, early in the day, I'd probably just go through it quickly, but it's, we're tired now, aren't we? That's 3, yeah? I think it's 7. I think they go around 7 times. 4, 5, Okay, and then back the other way. Okay, that's what it means to be in a low Reynolds number world. Everything is reversible. If you do something and then do it backwards the other way, you'll get back to where you started from. And that has various consequences for bacteria. Because if I'm a bacterium, okay, and I want to do a swimming stroke, so I'm trying to swim, so I do this, okay, and I move somewhere. But then I go back to where I was before, right, and I just end up back where I was. So any sort of stroke which looks the same forwards and backwards in time will get these little creatures nowhere. If you're a bacterium, somehow your swimming stroke has to be different forwards and backwards in time so that you can move. That's called the scallop theorem. And it's why bacteria have tails like this. All right? This thing wriggles its tail, and so there's a wave going round down the tail. And a wave has a direction in time. It will look different forwards and backwards in time. And so that's okay. This thing can move. Similarly here, this thing, I think this is the one which has this rotating motor there. And so its tail is going round and round like a helical corkscrew. And a helix has a direction to it. If you put your hand around the helix, it has a direction. If it goes around the other way, it would go the other way, other direction. And so this has a direction in time. And they have to be designed like that so that they can actually move through this fluid. What was I going to say? Yeah, okay, so that's the scallop theorem. Swimmers have to have a swimming stroke which is non-reversible in time to move. Oh yes, what I was going to say was if you want to try it out, what you have to do is fill a swimming pool full of treacle, molasses, what do we have in Brazil? What do we have in Brazil? Sorry? Molasses, okay, anyway, you know what I mean, golden syrup, that sort of thing. All right, jump in and try and swim. You won't be able to move because of this scallop theorem. You'll have to have some sort of non-time invariant stroke to get you somewhere. In fact, in terms of these, where's my, that's it. In terms of what I'm going to talk about, that's cute, but it's a bit irrelevant. There's another property of these equations which is actually far more useful for the active matter and far more important. And that is that these things are linear. Okay? Linear in V, they've only got a V in there. The nonlinear term went away when we got rid of the inertial terms. And that means I can add up various solutions to the Navier-Stokes equations, and it means I can do a multipole expansion for anyone who knows what that is so far. Yeah. And it means I can define a Green's function. So what I want to do now is try and understand what the flow field produced by a swimmer is. So I've got my little swimmer, little bacterium, moving through the fluid. What it's going to do is it's going to make the fluid move around it. Okay? It's like if you're in the bath and you sort of squish backwards and forwards, all the bath water is going to move. Or in a swimming pool, you know, someone in the middle is swimming, but the fluid all over the swimming pool is going to move. For these bacteria, what does that surrounding flow field look like? Well, let's start with something a bit simpler. Let's think about a colloid. And let's just take that colloid and pull it through the fluid. So there's a force acting on a tiny particle, and it's being pulled along here through the fluid. And the surrounding flow field looks like this. Makes sense, doesn't it? You're pulling this thing, and the flow field you'd expect to move along in the same direction with the colloid. And people know how to write down that flow field mathematically. If you're not used to the maths, it really doesn't matter. Look at the picture. But if you like the maths, here it is. Here's the velocity at a point r away from the swimmer and how it's related to the force on the swimmer. That mu is viscosity. This is the unit tensor. And r is just the vector from the point to the point where we're measuring the velocity. Or if you like index notation better, that's exactly the same equation written in index notation. And then this thing here is the Green's function. These are really words for the people who are sort of used to the math so they can see how it fits together. It's not very important if you're not used to it. This Green's function says, if I have a point force on this system, what's the velocity? And it looks like this, right? Okay. So now let's do the same thing for a little swimmer. Let's put a little swimmer here instead of a colloid and ask the same question about the flow field. So there's my swimmer, and it's exerting forces on the fluid, but it has to exert forces in equal and opposite pairs, okay? Let's think why. Well, there's nothing there to pull it, right? With the colloid, we had to pull it through the fluid. All right? There can be no net force acting on a swimmer because there's nothing there to pull it. It has to move itself. So from Newton's law, there can be lots of forces, but the most important one is going to be forces which are equal and opposite to each other. You can't have a force on its own. You always have to have these two equal and opposite forces. You have to have a dipole of forces. And so because that force distribution is different, we're going to have a flow field which looks different a long way away from this swimmer. Okay? So what I've done here is just say, look, this force is going to give me a velocity field like I had before, a distance r1 away. This force is going to give me a velocity field at a point here, which is a distance r2 away. Let's just add them up and see what I get. Add up the effect of two forces which are really quite close to each other a long way away. Let's see what comes out. And the answer is that what comes out is a flow field which looks like this, which means absolutely nothing, I think, with that formula, but let's look at the picture, okay? It's a flow field which looks like this. Here's my little swimmer in the middle. And what it does is it pulls fluid in at its ends and pushes it out from its sides. Okay? That's very different from what's going on here, right? Here, you have a symmetry where you have a special direction. Here, the symmetry is very, very different for the flow field around a swimmer. It's a symmetry which says that basically if I put a line down the middle, that side and that side are the same as each other, and that's called a nematic symmetry. Swimmers have a flow field, a far flow field, which has nematic symmetry. Also here, the flow field goes like 1 over r, and over there it goes as 1 over r squared. Think electromagnetism, right? What's that remind you of? Dipoles, yeah. So it would be like, it's very much like what you do when you do a multipole expansion for a point charge. A point charge gives you a potential 1 over r. If you have a plus and minus charge, that 1 over r bit is cancelled out, and you pick up the next leading order, which is 1 over r squared. This is exactly the same thing, but for forces instead of electromagnetism. Okay, let's just look and see if it really works by showing you the results and some very beautiful experiments, much as I don't like flogging very nice experiments from Cambridge. This one is a very beautiful experiment done by Ray Goldstein's group. What the poor graduate student did is measure the flow field around E. coli. And of course, this is a very noisy experiment. So he measured the flow field around about 500 E. coli and then averaged, very patient graduate student. And you can see that you have this symmetry again. This one's a bit different. This one pushes fluid out from the ends and pulls it in from the sides. Okay, but that's all right. That's just a change in sign, and we'll just look at that again in a minute. But again, you have this nematic symmetry, this special symmetry around the axis through the swimmer. Okay, so this is just words which we're going to use later on, okay? So you either have one which pushes it out from its ends. So here's my swimmer. It can push the fluid out from its ends and pull it in from the sides. And you call that extensile or pusher because this is the extended direction of the body and the flow field is trying to extend it even more. This one here, where it pulls fluid in from the sides and then pushes it out from the middle, is called a contractile swimmer or a puller because the flow is tending to squash it back to, it's tending to take it on its elongated direction and push it back in again. So let me summarize what I said, okay? So this is the important bit that I'm going to say now, which I need later on. Because swimmers are autonomous things, because they have no net force on them, because there's nothing there to sort of pull them, what it means is that their flow field, at least at long distances, looks like this. And what turns out to be important about it looking like this is that it has nematic symmetry. It has symmetry so that if I flipped it over like this, it would look the same. Okay? The flow field having nematic symmetry turns out to be very important when we're looking at these active nematics. Right, do people have questions about that? Any questions? Yeah.
Those experiments, we use power law or 1 over 2?
Yes. Yes, you get the power law very nicely. It's a far field approximation, so very close to, it doesn't work. And of course it couldn't, because if you just had one force like that and one force like that, they'd balance out and nothing would move. So it's a far field story. But yes, you do get the 1 over r squared. Yeah?
I'm not so used with the fluid mechanics field, but is this concept of dipole of force is well spread in the field? It's well known? Is that kind of old stuff? Or?
Yes.
Yeah.
Yes. I mean, originally I think it would have been done by the Cambridge lot who did everything 50 years ago. But it's only been used rather recently in this context of swimmers. Yeah.
And there are other laws of electromagnetism, also let's say Gauss, the law of Gauss or something like this, they are also applied here. I mean, if you have a closed surface, you can close this, you can get the direction of the field in that position, or just from the mathematical point of view that one goes over.
No, it doesn't. Because in one case you're doing Navier-Stokes equation, the other you're doing Maxwell's equations, and the analogy doesn't, there are some analogies, but it's not a complete analogy. What this is that because the equation is linear, you're allowed to do an expansion at large distances. You lose the leading term, so you're picking up the next term. Yeah, everybody else, right. So what I'm going to do in the, I mean, that was a slightly hand-waving version of this multipolar expansion. What I'm going to do in the class tonight, what you're going to do in the class tonight, is actually do it in detail, all right? So this is the proper mathematics of this multipolar expansion. I've got it all written out in one of the papers I sent you, and the idea is that you guys go through it because you won't understand it properly until you've gone through it. So people who are interested in that, the sort of level of mathematics you would need, I think, is to understand about index notation. If you're happy with index notation, there'll be no problem with that. If you're not, this is the version that perhaps it's best to stick with. So that's the class thing tonight. The other one will be much less mathematical. I've sent round 2 papers about motile topological defects in biology, and the idea is to discuss those 2 papers. But we won't have done topological defects till Friday, and so that will fit better then. So anyone who's signed up for this class who really doesn't want to do this maths, it's absolutely fine, isn't it, if they switch and go somewhere else, or vice versa. That's all right, isn't it? Yeah, yeah. But I mean, don't worry. Nobody's going to make you do it before you go home or anything. You know, we're all going to help each other, so it's all right. Yeah. OK, so the other thing, the other bit of background which I'd like to get done today is that word nematic again. People understand a lot about nematic liquid crystals. And the theory of active nematics is very much built on the theory of nematic liquid crystals. So let's learn about nematic liquid crystals. That thing is what I mean by nematic symmetry. A preferred direction, but it's not like a spin because up and down are the same. So a nematic is something which is made out of molecules or colloids of that sort of symmetry, right? Up-down symmetry. Same symmetry as we saw in the flow fields. At high temperatures, molecules will point every which way, but if you cool them down or push them together, they tend to align in this nematic phase. And the nematic phase is one where the molecules tend to all point in the same direction. And so there's orientational order. They're all more or less pointing the same way. But there's no positional order. If I looked at the center of those molecules, there wouldn't be any pattern to it like there is in a crystal. Okay? So this is a phase transition, if you like. Molecules completely isotropic at high temperatures. Cool them down a bit, they become nematic. Cool them down a bit more, they do all sorts of funny things which you're not going to worry about. Phase transition, so we need an order parameter to tell us how ordered it is. And there are 2 possibilities. One is a director field, which is just a vector in the direction of the molecules. And I notice I've done something very bad there. What have I done that's very bad? What's wrong with that? Go on, don't be scared. Tell me. I won't be crossing you. I might pick on someone if someone isn't brave enough to tell me. Go on. Ladies. Go on, tell me. Exactly. You knew exactly what it was. Don't be scared to yeah. Right, I've made it into a vector, haven't I? And there shouldn't be a preferred direction, there should be a preferred axis. So I should have a point on the bottom as well, or no point on the top. Okay, so that's one possibility for the order parameter. Another one is this thing, which is called the Q tensor. It's a tensor which is 3 by 3 in 3 dimensions and 2 by 2 in 2 dimensions. It's a symmetric traceless tensor, and that tells me a bit more. Okay, it tells me its largest eigenvector tells me the direction of these molecules, and its largest eigenvalue tells me how aligned they are. So it tells me a bit more about the ordering okay? But the details are perhaps not very important. So we've got this nematic described by an order parameter Q, and Q has 0 elements in the isotropic phase, and in the ordered phase, largest eigenvector is the direction, largest eigenvalue is how ordered it is. Okay, so this is the Oxford version of nematics. This is in the summer at high temperatures, where everything is isotropic, and that's in the winter at low temperatures in the nematic phase. These are punts. Okay, this is what you do in Oxford in the summer. You put on nice hats, and you get lots of champagne, and you punt up and down the river laughing at tourists who can't do it and keep falling in. Okay. For those of you that are used to Landau free energies, Landau expansions, this is how you write down the free energy of this nematic phase. These are the bulk terms where I choose all combinations of the order parameter, which gives me a scalar. And this just tells you about a phase transition from the isotropic phase to the nematic phase. If you aren't used to Landau expansions, don't worry. But worry instead about this. This is a term which says if I have any gradients in the order parameter, I have an extra contribution to the energy. And these pictures tell me why, okay? Here's my liquid crystal. Should have made them a bit bigger, sorry, right? And here I'm splaying out the order. It wants to be nematic. It wants everything to be lined up, and they're not, right? They're splayed out. And so there's going to be an extra energy because you're mucking up the nematic order. And this thing is called the elastic constant, and it tells me, you know, how bad it is to deform this crystal. In 3D, you can have different sorts of disorder. You can either bend them like that if you want to, or you can twist them round like that. So any deviation in the nematic order has an extra elastic energy. And that's the first reason these liquid crystals are cute. They're an elastic liquid. They're a liquid. If you look at them, they flow around, yeah? But they're elastic. The other reason that they're cute, and this is really important in this slide, okay, is that they have topological defects. Topological defects are places where the nematic order goes wrong, and it goes wrong in a big way because it's going to be wrong all the way out to infinity. So it's a bit like a knot. You can't undo the mistakes without changing things all the way out to infinity. In 2 dimensions, which is mostly what we're going to look at, because that's where the experiments are, the most important topological defects are this one. Okay, this looks like a comet, and that has topological charge plus a half. This one looks like it's got this threefold symmetry. Okay, threefold symmetry, and that has topological charge minus a half. And the topological charge is just a measure. It's just a way of classifying these things, really. If you take the line integral of the direction of the director field around the defect, here you get plus a half, and there you get minus a half. You can see this perhaps best by, if it's a plus a half one and you follow it around, look which way round my pointer's going. If you follow around the direction of that defect, it goes round like that. If I follow the direction of the director field, this one here, it goes round in the opposite direction. That's not super important, but what is super important is remember that shape, please, right? That's a plus a half. It looks like a comet. That's a minus a half, and it looks like a threefold symmetry thing, like a sort of flower. Okay? We're going to see these again and again, so if I could sort of, you know, get used to them now. In a perfect Nematic, you could never see these things because they'd have infinite energy, because they muck things up all the way to infinity. So they can't appear. You can't just have a plus a half defect or a minus a half defect, because they'd be infinite in energy. What you get is pairs. You take pairs of the things, plus a half and minus a half, because then you can have pairs, and a long way away you can get back to a proper ordered director field. And what will happen in any sort of real liquid crystal, any sort of normal passive liquid crystal, a normal sort of, like in your laptop liquid crystal, is that the plus and minus a half will gradually move towards each other and anneal out. Because then you get rid of these nasty distortions, which have lots of energy, and you end up with a nice, organized, nematic, perfect, ordered liquid crystal. So plus and a half, minus a half defects anneal out in pairs, just like charges. Okay? This is what happens in your laptop screens. People just spend their time trying to get rid of defects like this because they muck up the sort of viewing angle you get on your laptops. Right, so plus and minus a half defects. Sorry, any questions? I wasn't sure. Was that clear? Everybody, please ask if I'm not making sense, because I've looked at these things so often. It's easy when you've seen them a lot, and I know it's not first time round. Okay? Everybody happy with that? Plus and minus a half defects, and they anneal out in pairs, and you get rid of them. Of course, these topological defects are all over the place. Dislocations in crystals are topological defects. Vortices and superfluids are topological defects. Everyone's really worried about cosmic strings in the early universe. Those are topological defects. And now we're going to see them in biology, okay? They're all over the place in physics. Now we're going to, in a couple of lectures, see them in biology. I must be close. When would I start? Sorry, there. Right. Can I have just another? Yeah, I think I'd quite like to get this bit over. Is that all right? Yeah. Okay, and then I'll finish and we can do something else. Right. So, actually, no, I'll start with this next time. There's one little hard bit. I'll start with that next time, and then that's probably best, because topological defects is a really good place to finish. We're up to here. I told you I didn't change this slide because I didn't get there. I actually did this one first. Told you about the Reynolds number hydrodynamics. I've told you about topological defects in nematics. What we'll get to tomorrow is why on earth, I mean, low Reynolds number hydrodynamics, okay, but then it's not meant to be turbulent. What on earth has topological defects got to do with that? All right, and that's tomorrow. That's Friday's lecture. Okay, thanks very much. Hi, yeah.
So I have one question on my phone. So the question regarding numerics, how people usually today solve partial differential equations? Do you use some specialized software, or you write code yourself?
A lot of people, I mean, there are, for Navier-Stokes, they're commercial packages, very, very good commercial packages. We write our own codes, just using lattice-Boltzmann normally, plus finite difference type methods, so nothing very sophisticated, but other people do it.
Commercial packages meaning like MATLAB or something really specialized for Navier-Stokes?
It's called Fluent,
Fluent.
is one of them, yeah, because the car industry spends its time solving Navier-Stokes to look at flow around cars and air dynamics, yeah. Yeah.
And then I guess a small comment, you were mentioning string theory and things like that. So in your example with T and line, I think there is also a relativistic version of Navier-Stokes equations, right? It was formulated by Lambda back on time.
Yes, yes.
So you could have examples with nuclei colliding and flow of particles and things like that.
Yeah, I don't know anything about it, but I'm sure you could, yeah. Hello?
I understand the elastic liquid because elasticity is shown by solids. So how liquids are showing elasticity?
Yep. Viscoelastic liquids, for example, I'm trying to think of a nicer example, mucus, all right, I can't think when I say example. If you pull it, it will go back again and it will also flow. Silly putty will, that silly putty stuff, which will flow on long time scales, but is elastic on shorter time scales. So you can definitely have liquids which are both viscous and elastic. And it depends on the time scale. With these liquid crystals, what you have to have is boundaries, because if I have it between two boundaries and I force it to be nematic on these boundaries going that way and then do that, okay, the nematic is still trying to line up to the boundaries, which is why it's going to stay in that, it's not just going to flow. Yeah, so it's fine to be both. Yeah.
First, thank you for a very interesting lecture. Just about the flow fields. So you mentioned, actually the examples in the theorem declared the flow cells in the directions in the one side and outwards both sides. So, most bacteria are flow regular shape, and nuclei also have a specific direction. If you do the flow field for an eukaryotic cell, so an alien cell, which is going to be much more irregular in shape and direction.
Interesting question. Anything which is not next to a surface has to have this far flow field, which is dipolar, because it's on its own, it's got no extra forces. Mammalian cells sit on a surface, and that immediately makes it different, okay? Because essentially you've got, the cell is pushing itself against the surface. And has anyone, quite honestly, I don't know if anyone, people tend not to measure flow fields around that. And they've done it about, I would expect it to have a polar component, but I'm not sure about the relative amounts between the polar component and the dipolar component, all right? The actual shape I don't think is terribly important because we're looking at far flow fields here. Yeah, good question. I don't recall any experiments on that, but there must be some. Okay.
So let's make a very quick break.